that link. I don't know why it was broken. So I have same video, just by a different person. So I'll start by playing us that.
The contraction of a skeletal muscle generates the force necessary to move the skeleton. A contraction is triggered by a series of molecular events known as the cross-bridge cycle. In a skeletal muscle fiber, the functional unit of contraction is called the sarcomere. A sarcomere shortens when myosin heads and thick myofilaments form cross bridges with actin molecules and thin myofilaments. The formation of a cross bridge is initiated when calcium ions released from the sarcoplasmic reticulum bind to troponin. This binding causes troponin to change shape. Tropomyosin moves away from the myosin binding sites on actin, allowing the myosin head to bind actin and form a cross bridge. Also note that the myosin head must be activated before a cross bridge cycle can begin. This occurs when ATP binds to the myosin head and is hydrolyzed to ADP and inorganic phosphate. The energy liberated from the hydrolysis of ATP activates the myosin head, forcing it into the cocked position. A cross-bridge cycle may be divided into 4 steps. Step 1, cross-bridge formation. The activated myosin head binds to actin, forming a cross-bridge. Inorganic phosphate is released, and the bond between myosin and actin becomes stronger. Step 2, the power stroke. ADP is released and the activated myosin head pivots, sliding the thin myofilament toward the center of the sarcomere. Step 3, cross-bridge detachment. When another ATP binds to the myosin head, the link between the myosin head and actin weakens and the myosin head detaches. Step 4, reactivation of the myosin head. ATP is hydrolyzed to ADP and inorganic phosphate. The energy released during hydrolysis reactivates the myosin head, returning it to the cocked position. As long as the binding sites on actin remain exposed, the cross-bridge cycle will repeat. And as the cycle repeats, the thin myofilaments are pulled toward each other and the sarcomere shortens. This shortening causes the whole muscle to contract. Cross-bridge cycling ends when calcium ions are actively transported back into the sarcoplasmic reticulum. Troponin returns to its original shape, allowing tropomyosin to glide over and cover the myosin binding site on actin.
Okay, so hopefully that makes a little bit of sense. Okay, so picking up where we left off on Tuesday, we're ready to move on to talk about some different things associated with muscles. The first is what we call the length-tension relationship, which is exactly what it sounds like, a relationship between the length of a muscle and the amount of tension it can produce. Now, tension, when it comes to muscle, essentially is just another way to describe force. So how strong of a contraction can be produced by a muscle is affected by the length of the muscle before contraction. So I'll explain when we read through. Length tension relationship for skeletal muscle indicates how the forcefulness or tension of muscle contraction depends on the length of the sarcomeres within a muscle before contraction begins. So how short the sarcomeres are versus how extended and how long they are, how wide they are, okay, how big they are, okay. In other words, force is dependent upon the number of cross bridges formed by the actin and myosin molecules, okay. Cross bridges, remember, are the connection between the actin and myosin. And if you want to write this down, it's a simple, easy way to think of it. More cross bridges equals more force. Once again, more cross bridges, more force. So this illustration over here on the right is actually a graphing measuring force as a product of the sarcomere's length. Okay. We've got 1, 2, 3, 4, 5 points plotted. These 5 pictures are associated with those 5 points, and these descriptions are telling you what's the situation during all 5 of those points. Starting at point 1, you guys all hopefully agree with me that this is the shortest of the 5 as far as sarcomere length goes. Here's what the sarcomere looks like, where the Z discs are super close together. It's like what I just did with my model when I pushed it together. Okay, this is the shortest a muscle can go. It can't go down to 0 because that would mean it's flatter than a piece of paper. Okay, but it's quite short. And once again, this is in sarcomeres, so we're dealing with microns. Okay, at its shortest point, can we get it any shorter? No, it's already at its shortest point. So at this point, no force can be developed, and that's why it's at 0. Okay, because the Z discs are already so close together. Myosin and actin are, as you can see, smashed, and there's no way to shorten it any farther. Okay, taking that sarcomere and shortening it lengthening it just a little bit, drastically increases the amount of force it can produce. Shown here with point 2, myosin filaments are slightly stretched, and actin is no longer overlapping, allowing for the formation of cross bridges and thus force production. Okay, you stretch it a little farther, we reach peak tension, meaning we are maximizing the number of cross bridges we can form. We stretch it a little farther, same thing, point 4. We don't see any increase in force, and that's because we've already maxed out the number of cross bridges we can form. Stretch it to point 5, to point where the thick filaments and thin filaments no longer overlap. If they don't overlap, that means no cross bridges can be formed. And if there's no cross bridges that can be formed, there's no way to produce force. There's no way to shorten if they can't connect and the myosin can't pull on the actin. This picture, this graph, actually is from the original scientific research study that this was first demonstrated in. Pretty cool experiment that they did where they took a muscle from a frog and removed it, hooked it up to this machine that they could alter the length of the muscle to these 5 different points, looking at the 5 different lengths of sarcomere. And then they hit it with an electrical impulse that then caused it to contract. And then they measured the amount of force. So it's really cool. But a frog muscle that's not in a frog is different than a muscle that's connected to joints. Okay. Is it possible for our muscles to be in all of these positions? Really not so much. Our muscles and the way that our joints are organized really try to maximize the amount of force that we can produce so that we kind of stay in this area where our force is high Okay, so that's what this last sentence states. Muscles within the body are positioned within the joint to function near peak tension, generally cannot stretch beyond this, or I could also say, or shorten beyond this because of the limitations created by our joints. So it's not like we can operate with our muscles in this kind of state, but it affects how we lift things, how we move things, where we, position our elbows in a specific way when we lift something up, and that kind of stuff. So that's one way to affect force. We're going to get into other methods here. Last time we met, we talked about the importance of ATP not only in muscular contractions, but also in muscular relaxations. Okay. So ATP is very important. I want to talk about how we produce ATP in our muscle fibers. Reading through, a huge amount of ATP is needed when muscles are contracting. During muscular contraction, ATP is needed to power the contraction cycle, to pump calcium back into the SR, that's part of the relaxation process, and for other metabolic reactions involved with muscular contractions that we didn't really get into. There's 3 ways to produce ATP in muscle fibers. The first is through what's called creatine phosphate. Creatine phosphate is something we're going to talk about on this page. Okay, reading through. When at rest, muscle fibers produce more ATP than they need for resting metabolism. So we take that ATP, the excess ATP that is, and we use it to synthesize creatine phosphate, or CP. Actually, you can also write it phosphocreatine, PC. So you'll see it kind of some ways, CP, some ways PC, same thing. It is an energy-rich molecule only found in muscle fibers, and it's created thanks to an enzyme called creatine kinase that transfers that third phosphate on ATP to a creatine, thus forming creatine phosphate. Simple as that. It's just creatine with a phosphate on the end of it, so that's why we call it creatine phosphate. Okay. What is creatine? Well, it's a small amino acid-like molecule that we have in our muscle fibers. We do this once again at rest to basically stockpile ATP, but we're not actually stockpiling the ATP itself. We're stockpiling this. That way, when muscular contraction begins, we can reverse the reaction, take that phosphate from creatine phosphate, give it back to ADP, and form the ATP that can then be used for muscular contractions. This process is described as being anaerobic, which is a term we learned about earlier in the semester, meaning it does not require what? It doesn't require not ATP, but oxygen. It's an ATP producing process. No oxygen is necessary to be formed. Creatine phosphate and ATP can provide enough energy for muscles to contract maximally for about 15 seconds, which is like super high intensity, short duration events or athletic events, activities like a 100-meter dash, set of lifting weights, okay, where you're doing something really, really powerful in a very short amount of time. Now, 15 seconds is not a very long amount of time, and if you're going to continue exercising at a maximal intensity, really, really hard, as you can, for longer than 15 seconds, you have to turn to other sources. That's where we turn to ATP from anaerobic cell respiration. Since it's called anaerobic cell respiration, this tells us right away that we do not require oxygen. This is what we do once again after we deplete the supply of creatine phosphate within our muscles. It involves the movement of glucose into the contracting muscle fibers, where we break it down in a series of 10 reactions called glycolysis. And thinking back to our lecture on the endocrine system, we had 6 big buzzwords. Glycolysis was the first that I think I typed it up. Where it's the breakdown of glucose to form ATP. This takes place in a cytosol, so it's a cytosolic reaction. It actually requires a little bit of energy input in the form of 2 ATP before we form a small amount 4 ATP. So actually, during this process, we only net out a total of 2 ATP because it's like putting 2 quarters in a slot machine, pulling the deal, and 4 quarters pop out. You don't win $1, you really win how much? 50 cents, right? Okay. At this point, we either form, well, we're going to definitely form pyruvic acid, 2 of them, but depending on the state of the cell, we will either use that pyruvic acid in the mitochondria for aerobic respiration, or it can be converted to lactic acid if we don't have enough oxygen in the blood, which is what this bullet's all about. So depending on the availability of oxygen in the muscle fiber, the end product, what we call pyruvate, same thing as pyruvic acid as it's labeled here, will enter the mitochondria for a series of ATP making steps that require oxygen, or the pyruvate will be converted to lactic acid, which, as it shows right here, will diffuse into the blood, which can drastically reduce the pH of the blood. Now, lactic acid is something that a lot of times gets blamed for a lot of things, especially if you like to watch sports. You'll hear announcers that have no real true background in physiology. They see a guy cramping up and they say, "Oh, he's got lactic acid." Or they've got an injury and something. They go, "Oh yeah, he's got lactic acid." Oh, no. Okay. Lactic acid does do some things. It is known to cause fatigue. It's not what causes cramps. It's not what causes muscle spasms, that kind of stuff. It just gets blamed by people that really don't know what they're talking about. It kind of drives me nuts, but that's because I know that it's not involved. So, lactic acid, not necessarily a bad thing all the time, but it does, cause fatigue, which we'll talk about fatigue here in a second. This type of ATP production is going to power activities when we are doing maximal muscle contractions, so working out as hard as we can for about 30 to 40 seconds. Examples of things that are about that long would be running a 400-meter race, 1 lap around a track. Most people are not even going to get anywhere close to all the way around in 30 to 40 seconds, but the majority of the race will be utilizing this form of ATP production. And as you guys are all very aware, there are athletes out there that run for a lot longer than 30 to 40 seconds. And that means they're going to have to then turn to something else to get their ATP. From aerobic cell respiration, which aerobic tells us that it requires oxygen. And this is what takes place in the mitochondria, which is why we call the mitochondria the powerhouse of the cell, but only occurs if sufficient oxygen is present, allowing that pyruvic acid that we just formed at the end of glycolysis to enter the mitochondria, where we then can oxidize it. We'll learn what that means later on in the semester, which ultimately produces a heck of a lot of ATP, but this is where carbon dioxide that we breathe off comes from. We'll actually form a little bit of water and a lot of heat as well. From an original glucose molecule, we can form about 36 molecules of ATP. When you count in the glycolysis 2 ATP that we form, along with some other stuff. Fatty acids, which we get from fats, like triglycerides, can yield more than 100 ATP, which is why fats are so much more higher in calories than carbohydrates. ATP produced during aerobic cell respiration provides enough energy for prolonged activity, provided sufficient oxygen and nutrients are available. In activities that are longer than 10 minutes, about 90% of ATP is used, or ATP used is provided by aerobic cell respiration. In super long, really long endurance athletic events like marathons that take hours, we're pretty much at 100% aerobic respiration. Now, later on in the semester, we literally have an entire lecture dedicated to cell metabolism and heat balance. That will be in unit 5, because this is unit 4, right? Whatever unit is, this is the next unit. No, 2 units down the line. So unit 6, I guess. We will talk all about every single thing here on these last 3 slides. We'll get into greater detail about using creatine phosphate for ATP, not much, but we'll definitely talk about glycolysis for sure in great detail. Talk about this, all the steps like the Krebs cycle, electron transport chain, which is what this is going to involve. As you can see right here, we'll even talk about how we can use proteins to make ATP, how fats are utilized to make ADP. And so we'll just kind of simplify things for now and just kind of introduce it. This next figure is actually from an exercise phys textbook that I use for that class, which is a class that some people take after they take this class. Just kind of breaks down on a nice, easy way to understand the comparison between aerobic and anaerobic. For time, as far as seconds to minutes to hours, 120 minutes obviously is 2 hours. And then I gotta have a lot of athletes on campus. Like I get a lot of softball players, volleyball players. I always kind of like to point out, to them where their sport ranks. Some sports are kind of tricky, like soccer, for example. You got different positions, like a goalie. Their job is really to kind of stand still at the goal, and then they have to like sprint. So it's kind of anaerobic, whereas like the midfielders are running all over the place. So it's not always 100% perfect, but it's kind of cool. Nothing I'm going to ask you guys to know for the exam now.
Okay, wait, what about cross country? Where's that?
Cross country, well, it just kind of depends. Cross country, what in high school you were like, 3 miles to 5K or whatever, which, is definitely 1,500 meters. That's what's just short of a mile, right? That's already at 70% aerobic, 30% anaerobic. So anything farther than that, there's cross country there, 90-10. I don't know if that's, for 3 miles or 10 miles or what, but it's, I guess, 10,000 meters, whatever that is. So. Hang on, somebody's knocking. Thank you. All right, as I promised you guys, I would talk a little bit about fatigue, something that affects our ability for muscles to contract. Muscular fatigue is described as the inability of a muscle to maintain force of contraction after a prolonged activity, like running 10,000 meters or 2 hours or whatever it is. There's actually kind of 2 different forms of fatigue, central and peripheral. Central fatigue is that feeling of tiredness and a desire to cease activity prior to actually feeling true muscle fatigue. So it's like when you're, out for a jog and you've been going for a little while and you just feel tired and you're like, okay, this sucks, I want to be done. Are you completely to the point where you can't go anymore? No, but you feel like you should stop, right? Okay, and this is where, you have a lot of intestinal fortitude. You're going to continue on past this central fatigue. We call it central fatigue because it's caused by changes occurring in the central nervous system. So within the brain and spinal cord, the exact mechanism isn't really known, but it may be protective in that it's going to stop a normal person from exercising before muscles start to become damaged. Peripheral fatigue involves factors after what we call the neuromuscular junction in MJs, where a neuron and a muscle meet. This impairs the force generating capacity of a muscle, and this is what we would describe as muscular fatigue, and that most evidence points to it as where things are starting to go wrong. Let's talk about those things going wrong. Although the precise mechanisms that cause fatigue are still not 100% clear, there are several factors that are thought to contribute, which includes inadequate release of calcium from the sarcoplasmic reticulum, which, as we know, without calcium, there's not a shift in tropomyosin, and thus there's no cross bridges. And as we just learned with the length-tension relationship, cross bridges affect force, so therefore it affects fatigue. Depletion of creatine phosphate, especially in those really, really short duration, high intensity activities. Insufficient oxygen, obviously that's going to play a role in, ultimately making the ATP, which is why, one of the reasons why it's more difficult to exercise at higher altitudes. Now, something else that you hear a lot of times when you watch sports is like, when the Chiefs go to Denver and they play, and the announcers say, well, there's less oxygen in the air. We're going to talk about why that's a really kind of terrible way to describe it. Here, when we talk about breathing and oxygen and that kind of stuff, depletion of glycogen, our stored form of glucose, as well as other nutrients. Basically, this is kind of like running out of gas, which in athletics is described as hitting the wall. Anybody heard that phrase before? I'll show you a video clip on that here in 1 second. I did say lactic acid is blamed for a lot of things, but I did mention that it does contribute to fatigue. A lot of that's because of what it does to our hydrogen ion concentrations. Hydrogen ions, remember, are associated with acids like lactic acid, and hydrogen ions can cause fatigue, typically only taking place during heavy exercise. And then a failure of action potentials in the motor neuron to release enough acetylcholine, because that's where it all began, as we learned the other day. All right, let's watch our video here on hitting.
Do you say this kind of stuff when you're watching sports? I feel like I can just see your wife being like, oh my God, it's okay. That's
sometimes.
What they say.
Sean Welch is in a desperate spot. Her body has run out of fuel. No calories to burn. Systems shutting down. The legs are there, you just can't feel it. The eyes still see, but through a gauzy veil of delirium.
So this is at the very tail end of an Ironman competition.
Is aware of two things.
And if you're not familiar with an Ironman, it's insane what they have to do.
Someone is so close behind. After more than 140 miles, it's come down to less than 100 feet.
Now, it's not 140 miles straight running. It's biking, swimming, and then running. I don't know if that's the correct order, but I do know the last leg is a marathon, 26.2 miles of running after 100 and whatever miles of biking / swimming. So most people are not doing this, only extreme individuals.
To that damn line. Just yards behind, her stalker appears. It's Wendy Ingram.
Okay, they kind of just talk. I'm not going to finish the rest of it. But like I said, this is essentially the point, hitting the wall where they are just out of energy substrates. They just are running out of energy. Have you ever been in a car where it's running out of gas? It starts to sputter, and it's like when it lunges and it's not hitting like it's supposed to. So you could see that's what was happening to those poor ladies. Now, obviously, you know, if you're just going out for a 15-minute jog, you're not going to be experiencing something like this. This is extremely long duration, high intensity type activities. I enjoy watching The Office. I've seen it many times. There's an episode where they're getting ready to run a 5K, and the main character's shoving in pasta. He's eating right before the race, which is absolutely not a good time to do it. But for a 40-minute or however long it takes that person or a normal person to run a 5K, they're not going to need to be doing that. Speaking of that, I'm talking about is called carbo loading. Anybody heard that phrase before? Carbo loading is one way that people try to help gain an advantage prior to a competition, like a race. A lot of times people carbo load when they really don't need to, but that's not something I'll get into. Carbo loading is a way of improving performance, or at least thought to improve performance, which is what an ergogenic aid is said to do. Ergogenic aids are defined as work-producing substances of phenomena believed to increase performance. Okay. Why is carbo loading actually beneficial? Well, it will increase your glycogen levels in your skeletal muscle. So it's like filling up your car tank, your car's gas tank all the way to the top before you go for a long drive. You're going to be able to drive farther with a full tank than you would if it's a half a tank, right? Glycogen is just a stored form of glucose, so it's just a stockpiled form of ways to make ATP. Okay. Earlier we talked about making ATP from creatine phosphate. Okay. You can increase the amount of creatine in your body, which thus means you can store more ATP and thus use that ATP later on during exercise. This is done by supplementing with what's called creatine monohydrate. A lot of times people just call it creatine. I imagine a lot of people have heard of creatine supplementation before. And it is beneficial because it improves the ability to maintain force and power output. What type of activity do people that supplement with creatine usually participate in? Who are the people that take creatine? What kind of athletes?
Runners.
Not runners,
Are you
typically.
talking about like bodybuilding?
Exactly, weightlifters, because
Yeah.
ATP from creatine is good for not very long, like a set of lifting weights. And they might be able to lift one or two more reps, which means they might get a little bit stronger. What would carbo loading be good for? Not lifting weights, running, like long distance, not like the 3 miles that Michael Scott was running in The Office, but something more long term. Or if you're training really hard, successful days in a row. Okay. Caffeine, I always joke, is one of college students' best friends, one of my best friends. Something you can get simply by, just driving down to Casey's. Can do a lot for us. May improve performance not only at the muscle level, but also at the nervous system, or even just delivering fuel to muscle. It can elevate blood glucose, increase fat utilization, which means more ATP, and just makes you feel less tired. Decreases perception of fatigue, lower rate of perceived exertion, so it doesn't feel like it's as hard to exercise. So an ergogenic aid can be something that you go and buy at GNC, like creatine. Could be something like you can get in a box of macaroni, carbo loading. Could be just something that you get in a 5-hour energy. Actually, it could be even simpler than that. Stretching that people do before an exercise event, before they run. because it makes them feel loose. They can run faster. Stretching actually can be described as an ergogenic aid. Breathing a higher percentage of oxygen is thought to also improve performance in some situations. Could be also considered an ergogenic aid. So really, it could be a lot of things. It could be like listening to music, firing yourself up. So
Hey, the full distances for Ironman, it's 2.4 miles of swimming, 112 bike ride, and then 26.22 running.
Yeah.
In that order. You're supposed to complete the swim within 2.5 hours.
yeah, it's an all-day event for sure.
Yeah.
And those people that do those, like the ladies that were there, they would have to be not just drinking water and Gatorade during the event. They'd have to, like Gatorade makes, I think they're called like G-Packs or something like that. It's basically just like sugar that you can just squirt into your mouth.
No, it's a different thing, but yeah.
They'd have to be eating food or, drinking something with lots of fuel reserves in it. Anyway, moving on. What's up?
I was kind of thinking about this. So it talks about like, where does it, the central fatigue is like to stop a person from exercising too much before the muscles become damaged. So if they're doing those kind of marathons all the time and they're doing that to their body all the time, that's gonna cause damage because they're getting stopped.
I mean, it can, but
often, or
but as you got to remember, the body's really adaptable, and that it can get used to, because we respond and we change the structure of our muscles. We make our bones get harder, our tendons get stronger, those kinds of things. So training goes a long ways. That's something I talk about in that muscle phys class, where I get into a lot of this stuff in greater detail. All right, moving on. Something I just mentioned back on that fatigue slide was the abbreviation NMJ. NMJ is short for neuromuscular junction. And I want to talk about that here on this page. So reading through, even though each skeletal muscle fiber has only a single neuromuscular junction, axons of a somatic motor neuron branch out and form neuromuscular junctions with many different muscle fibers. Like what we show here with a purple motor neuron and a green motor neuron, how the purple is innervating looks like these two muscle fibers. The green is going to innervate these three muscle fibers. A somatic motor neuron and all the muscle fibers that it innervates is what a motor unit is. Okay. In other words, it's the neuron plus the cells that it causes to contract. Okay. It can be as few as 20 muscle fibers, can be as many as 1,000, depending on what type of control is needed. When a nerve signal approaches the end of an axon, so when an action potential arrives at those distal branches, an action potential is going to spread to every single one of those synaptic bulbs at the same time. And so each muscle fiber becomes excited at the same time, and they all contract in unison, all together at once. So if a nerve signal comes down this green one, this muscle cell, and that muscle cell are all going to contract at the same time. If an action potential is coming down this purple one, and that one are going to contract at the same time. And this morning in anatomy, we talked about the extrinsic eye muscles, the ones that move our eyes. I said they provide the finest motor control in the body. This is what I was talking about. Fine motor or muscular control requires small motor units where there's just only 20 or so muscle fibers per neuron. So it's very, very coordinated, very, very precise, and very, very pinpointed. We can, direct our eyes in a very, very specific manner. We also have great fine motor control in muscles in like our hands, our fingers, our wrist, to allow us to write some just better than others. A strength motor muscular control is going to produce a big, bulky muscular contraction, very strong, large scale, powerful movements, because you're going to end up contracting 1,000 or so muscle fibers at once. So very, very cool. Now, why I talk about that? Because it will play a role here in just a little bit. Let's talk about controlling muscular tension. All right, reading through. A single nerve impulse in a somatic motor neuron elicits a single muscle action potential in all the skeletal muscle fibers with which it synapses with. Action potentials always have the same size in a given neuron or muscle fiber, meaning no matter how strong the stimulus, we depolarize, to that positive 30, 35 number before we repolarize, right? Talked about that when we talked about neurons, same thing in muscles. However, in contrast, the force of a muscle fiber contraction can vary, meaning muscle fibers can produce much greater force than they do if you just have a singular action potential. Total force or tension that a single muscle fiber can produce depends mainly on the rate at which nerve impulses arrive at the neuromuscular junction. This number of impulses per second is called its frequency, okay? Which we've talked about frequency before, demonstrated here on this page where we show action potentials in blue here. Really close together, this we would say has a high frequency, a little bit more spread out as a lower frequency. Look at the difference in strength of contraction or force of contraction here versus here. I'll get into that more here in a second. So that's what frequency of stimulation refers to. The more often we send an action potential to a muscle fiber, the stronger its contraction will be. Okay. Now, a twitch contraction is what we're showing here on this page, is a brief contraction of all the muscle fibers in a motor unit in response to just one action potential. This is not something we do naturally, because naturally we send a bunch at once. We just don't always send them at high rates. Sometimes we send them more at a low rate or frequency. So in other words, this has to be done in a lab setting with direct electrical stimulation, like hooking up an electrode to the muscle, there's other ways to do it, but that's really the best way to do it, which can either be done on the neuron itself or the muscle fibers. Yeah. Anybody ever been hooked up to a e-stim machine? One that you put little electrode pads on your arm or leg or whatever, and then they turn a dial and it sends electrical signals into your muscle. It can cause them to contract. It's a thing that's done in like physical therapy. But if you crank the machine all the way to the right, crank up that, it's going to cause the muscle to go into tetany, which I'll talk about here in a second. So here's what a twitch would look like. There's kind of three major periods to it. The first is its latent period. Whenever you see the word latent, it's not the first time we've seen that. Just think delayed. Now, this is a very short delay. We're talking milliseconds here, just like we saw with neurons. There's not much of a delay, but this is the period. It's about 2 milliseconds in which there's the delay between the stimulus, where we hit it with the signal, before we see any muscle tension developing. So there's no contraction taking place at this point. And it's because the action potential is sweeping over the sarcolemma and we're releasing calcium from the SR. And then we develop tension during the contraction period, which can be as fast as 10 or as slow as 100 milliseconds, which sounds really weird to describe 100 milliseconds as slow. But if you just think about it without the M in front, it might make more sense. 10 seconds versus 100 seconds. 100's a lot slower, right? Okay. Same thing when we deal with milliseconds, although 100 milliseconds is still fast. It's just not nearly as fast as 10 milliseconds. This is where the calcium's binding to troponin, myosin binding sites on actin are exposed, cross bridges form, and the shortening occurs. We develop peak tension during this stage. And then, since it's only one action potential, we relax, meaning that there's calcium being pushed back into the SR, myosin binding sites are being covered by the tropomyosin, myosin heads are breaking free from the actin and can't reform cross bridges, and our tension is going to go away. Now, this graph does not show the refractory period, which we saw refractory periods when we talked about action potentials, and how I kind of use the analogy of you can't jump a second time until you land the first time, right? Okay. When a muscle fiber contracts, it temporarily cannot respond to another action potential, kind of like what we talked about with neurons and action potentials, but it's a little bit different, and we'll talk about that on this page. Okay, like that of nerve fibers, when a second stimulus occurs in a skeletal muscle after the refractory period of the first stimulus is over, but before the muscle fiber has relaxed, a second contraction will actually be stronger than the first. So, looking at our graph down here at the bottom, I've already kind of pointed out, but the blue lines down here at the bottom represent the action potential in the muscle fiber. Measured up here in red is the force of contraction. If we show this first area, we hit it with one action potential, it contracts and relaxes. That's what we showed on the previous page, a twitch. Okay. Over here in our second portion of the graph, we call this wave summation, where we show two action potentials. The first causes contraction, and as you can see, it's the same amount and same strength of contraction as what we saw over here with twitch. As we start to relax, before we relax all the way to rest with no force, we hit it with a second action potential, which causes force production to go up even more. And so summation, we talked about summation in neurons. And I said summation, the beginning of that word spells out the word sum, which in math you calculate what when you calculate a sum? Addition, right? Okay. So literally the additive, add is in the word additive, effects of contractions of different muscle fibers, causing a second contraction being greater than the first. Okay. And this continues on into the third region of our figure, where we have what's called incomplete or unfused tetanus, where we hit it with more and more and more action potentials, thus causing force to continue to go up. So this occurs when electrical stimulation occurs so rapidly that the relaxation time between the twitches becomes shorter and shorter and shorter, and the strength of contraction continues to rise. Is it going to rise on forever and ever and ever? No, because 0.5 tells you and shows you that eventually we'll reach a point where we max out the force of contraction, which should make sense because our muscles aren't infinitely strong, are they? Okay. Complete or fused tetany, or tetanus, occurs when stimulation allows for no relaxation. So we don't see the dipping off like we show here. All we see is a contraction appearing smooth and sustained until we hit this point when it's just basically a held contraction. Now, tetanus is a word that you've probably heard before, and it's actually something we've talked about before back in that nervous tissue lecture. We talked about tetanus toxin, how like if you don't get a tetanus shot, you get that toxin into your body and it can cause your muscles to lock up like lockjaw, right? Okay. this is what happens when somebody has tetanus and doesn't get that tetanus shot. Okay? Their muscles are going to end up sustaining a contraction, like in the muscles of their jaw, causing it to lock up. And I was just talking about electrical stim machines used in physical therapy programs. And I took a class where we got to play around with them. We were learning about rehab stuff. And there was a lab we had to do where we basically caused muscles to go through this by cranking the machine all the way up. And it hurt. We didn't do it for a long period of time, but it did not feel very good. Yeah, not quite, but also probably not a fun experience. Okay, another thing that affects force production relates back to motor units, which I said we'd refer back to again. Once again, I'll bounce back to the page where we introduced what a motor unit is. Motor unit, remember, is a somatic motor neuron and all the muscle fibers it innervates or stimulates. So it's like this green neuron plus these 3 muscle fibers. Okay. So in motor unit recruitment, we are recruiting or increasing the number of motor units used. Like what this refers to is the fact that when we do a muscular activity, more than likely, it's not going to require 100% of the muscle fibers to be done. Like writing your notes, picking up the pen off the table, does it require a maximal contraction? Absolutely not. Okay. We only need to use as many muscle fibers as we need, right? Okay. So reading through, typically different motor units of an entire muscle are not stimulated to contract in unison. While some motor units are contracted, others are relaxed. And this pattern of activity delays muscular fatigue. Because think how easily tired we would get if we, every time we did anything, contracted every single muscle fiber in every single muscle. It would be exhausting and we would not be able to get much done. Okay. So this allows contraction of a whole muscle to be sustained for longer periods of time. We're going to recruit the weakest motor units first. Once again, we're measuring tension or force. If our force or tension needed is not much, we recruit our smallest muscle fibers, our smallest motor units. If we lift something a little bit heavier, we're going to recruit a few larger muscle fibers. And if we recruit all of them, it's only because we require a lot of force or tension. And this is one factor that also helps with producing smooth muscular movements rather than a series of muscular jerks. Not jerks like your mean jerks, but like herky-jerky motion so that we are not bouncing around as we're picking stuff up or moving around or whatever. Something that we've also talked about before, thinking back to the nervous system, was this concept of muscle tone, which states that even at rest, a skeletal muscle exhibits muscular tone or muscle tone, a small amount of tension in our muscles due to weak involuntary contractions of its motor units, which kind of goes against what we've been talking about, and that skeletal muscles are voluntarily controlled, but there are also these involuntary muscular contractions taking place in our muscles, even when we feel like they are nice and relaxed. It is established by neurons in our brain and spinal cord, resulting in excitation of the muscle's motor neurons that cause the release of small amounts of acetylcholine. when motor neurons that serve a skeletal muscle are damaged or cut, like in a spinal cord injury, muscles become flaccid, something hopefully we never experience, a state of limpness in which muscle tone is completely lost. At that point, muscles cannot contract without an electrical stimulation method from outside the body. To sustain muscle tone, small groups of motor units are alternatively active and inactive. Alternatively, not alternatively. Okay, let's talk about the two types of muscular contractions that we can perform. And it all relates to the length of the muscle during the contraction. There is what we call isotonic contractions and isometric contractions. Isotonic contractions are where tension or force of contraction developed remains constant. And here's the key. While the muscle changes, and I italicize that word, changes its length. This is what we typically would think of when we talk about muscular contraction. It causes movement, like picking something up, like a book that our fella in the pictures is doing. Involves two phases, concentric and eccentric. Concentric, I would highlight the term shortening. So when he's lifting the book up, if we pretended that was a dumbbell instead, he's lifting the barbell dumbbell up and his biceps is shortening. Reducing the angle of the joint and then coming back down, lengthening the muscle. I should say lengthening, not length. Well, I guess it is right the way it is written. Involve the length of the muscle increasing. So length increasing would be important words to highlight there. When you see people in a gym, I used to work in one when I was in college, you see people that lifting weights, kind of you can tell they've never done it before. One thing they don't always do a good job of is taking care of the eccentric phase of a muscular contraction. A lift can be done where you bring the dumbbell up and a bicep curl and you just drop it. And you're really taking out a really important aspect of that lift, which is why a lot of times you see people that really kind of know what they're doing, kind of taking that barbell down or dumbbell down kind of slowly, where actually a lot of the gains that you get, the beneficial parts of a lift come during that eccentric phase. Walking down a hill, for example, is going to cause you to be a little bit more sore a lot of times because you're doing a lot of eccentric contractions and it's kind of harder on the muscle, which is what causes you to feel a little bit of that soreness. Isometric contractions, key is we're generating force, but and as you can see, I italicized the muscle's not changing its length. Like holding a book from an outstretched arm. I always talk about wall sits. I do a lot of these when I was rehabbing my knee when I was in college after I tore a ligament. I had to do a squat up against a wall and just hold it for like a minute or so. If you've never done that, try it out, and you'll feel after 10, 15, 30, 45 seconds, your legs feel like they're about to catch on fire because it hurts. You're producing a lot of force, but your muscle's not changing its length. That type of motion is not as prominent in athletics. It's done a lot in like gymnastics. Like you see the people holding a position, dance probably a little bit as well. But more so as far as athletics and physical activity, you're doing a lot of this shortening and lengthening because it's motion a lot of times, not staying still. So everybody good on the differences between the two? Okay, this is a perfect spot to stop, especially since we are out of time. So we will stop here. What I'm going to do, I'm pausing it right now, but I'm going to just pick up with this as soon as you guys are all gone, and I'll finish from here. So, all right, and thanks to the power of technology, you didn't even know I was taking a break unless you were in class. So we will pick up right where I was just at. Just finished talking about different types of muscular contractions. We're going to switch gears here now to talk about what we call muscle fiber types. Remember, muscle fibers are the cells of a muscle, and within our muscles, the skeletal muscle fibers are not always going to be 100% alike. They differ in some different ways: composition, so what they're made up, or how they're structurally made up, and how they function, which demonstrates the relationship between structure and function, anatomy and physiology. One of the important things that muscle fibers differ in is their content of myoglobin, something that we talked about very early in this lecture when we talked about muscle fibers was myoglobin, a protein in our muscle cells that helps bind and store oxygen, which relates to ATP production. We have what we call red muscle fibers and white muscle fibers. Red muscle fibers appear red because they have a high myoglobin content, and this is actually what causes dark meat in chicken, turkey, and those kinds of animals that we have a lot for food purposes, and in my opinion tastes better. Red muscle fibers contain more mitochondria. Mitochondria, remember, is where we use oxygen to make ATP. And then they're supplied by more blood capillaries to deliver that oxygen to the myoglobin that can then be used for making ATP. White muscle fibers are going to have a much lower content of myoglobin and are going to appear like the white meat, appear lighter in like chicken breast, for example. So those are kind of their just like easy pinpoint ways based on what they look like. There's also ways to describe them and break them down into what we have down at the bottom, type 1, type 2A, and type 2X, which relates to several other factors: how fast they contract and relax, what type of metabolic reactions they use to make ATP, how quickly they fatigue. So those are all factors that we'll be talking about here on the next several slides. Next, I think 3 slides. The first type I want to talk about are called type 1. That's kind of the traditional way to describe them. Your textbook lists them as slow oxidative fibers. Definitely know them both ways. Slow oxidative is a nice descriptive way of describing them, in that they are not quick in contraction speed. Oxidative refers to the fact that they make their ATP using oxygen, what we call aerobic cell respiration. I learned them as type 1, so like I said, know them both ways. These are described as being smallest in diameter, and that means that they are the least powerful type, because the bigger the diameter, the stronger of a contraction they produce. They appear dark red because they have a lot of myoglobin, contain a lot of mitochondria, which enables them to make their ATP mainly by aerobic cell respiration. They're slow, as it says there at the top. Their twitch contractions can last as slow as 100 to 200 milliseconds, which, remember, slow is very relative here because 100 and 200 milliseconds still very fast, but much slower than what we'll see here with our type 2X fibers. Even though they are slow, they're very resistant to fatigue, so they don't get tired very easily, which is going to help make them capable of prolonged sustained contractions for many hours. And this is what you'll find a lot of in what we call our postural muscles that are like up and down your spine, allowing you to keep your head straight up and down. And they're also found in high quantities in endurance athletes, those that are really good at running marathons, people that are good at just continuously exercising for hours and hours and hours, in a not super intense manner. I'm going to skip ahead actually to talk about these next type 2X. I'll go back to the type 2As here in a second. Type 2Xs are described as being fast glycolytic. Fast because they are very fast, contracting quickly and strongly, as it states here. Glycolytic refers to the fact that they make their ATP by glycolysis, which we learned earlier is an anaerobic form of ATP production. They are the largest in diameter and generate the most powerful contractions. Basically, these are the exact opposite of the type 1s, which were really slow and oxidative. Small diameter, not very powerful. These, on the other hand, are going to contain a lot less myoglobin, a lot less blood capillaries. They aren't going to bring in as much oxygen because they make their ATP by anaerobic means. Okay, they don't need as much mitochondria because glycolysis takes place in the cytosol. Now, does that mean that they don't have any mitochondria, that they don't have any blood capillaries, they don't have any myoglobin? No, they just don't have as much as the type 1s. Type 1s appeared red, these guys appear white. Once again, they contract really fast, really strongly, but they also fatigue really quickly. Type 1's really good for long-term, long-term aerobic endurance activities. These are really adapted for intense anaerobic movements of short duration, like weightlifting, throwing a ball, what we'd like to call like the power sports. Football players are going to be loaded up with type 2 fibers, type 2X fibers, really strong, really powerful, but for a very short amount of time. Sprinters, I always say Usain Bolt, who is the world record holder of, I know, at least the 100 and the 200 meters, track sprinter from Jamaica, incredible athlete, fun to watch, just is probably loaded up with type 2 fibers, type 2X fibers. And then in the middle, kind of intermediate between the type 1s and type 2Xs are type 2As, which are described as fast oxidative glycolytic, meaning that they are going to contract in a fast manner. Oxidative means that they make their ATP by aerobic respiration. Glycolytic tells you that they also generate ATP anaerobically through glycolysis. Once again, I use the term intermediate, and that's because they're somewhere in the middle between the other two types in things like diameter. They're kind of a hybrid between the two, containing large amounts of myoglobin, many blood capillaries, allowing them to make their ATP considerably by aerobic respiration, dark red in appearance, moderately resistant to fatigue. Once again, do generate some ATP anaerobically. The speed is faster in these guys than it was in the type 1s, not as fast as the type 2s, 2Xs, I should say. And they're going to contribute to activities, walking, running, sprinting, that kind of stuff, as you see here. Table 8.2 is another thing I snagged from my exercise physiology textbook, kind of give you a breakdown on percentages of distance runners, what they have in their body. For muscles, it's about 70 to 80% type 1, 20 to 30% 2X and 2A. Track sprinters are the opposite, a lot less of the type 1s, much more of the type 2Xs and 2As. For non-athletes, the general population is pretty well 50-50. Now, thinking about this, most of all of this, as far as being a high-level distance runner, high-level track sprinter, whether you're more adapted to one or the other relates a lot to, and predominantly, your genetics, which means, mom and dad. So stuff we cannot control. You can train and do different things that will affect the percentages, but really it's pretty minimal. So if you want to be a world-class sprinter or a world-class distance runner, as they say, you better pick your parents well, which obviously we can't do. Okay, moving on from fiber types, I want to talk about the neuromuscular junction here again for the, I don't know, 4th, 5th, 6th time so far here in this second half of the lecture. So reading through with all the processes taking place at the neuromuscular synapse or the neuromuscular junction, it is not surprising to find it vulnerable to drugs and paralytic toxic agents. Something we talked about back in our nervous tissue lecture was botulism toxin, or Botox as we've described it before, which we talked about is found in improperly canned goods. So if you are shopping, don't get the ones with dents in them, or uses a medicine called Botox. What it does is it blocks the release of acetylcholine from synaptic vesicles. So it means that neurons are not going to cause excitation in the muscles, which means that the muscles cannot contract. And that's what people do when they inject the Botox in their face, causing their muscles of the face to completely relax and thus take away their wrinkles, or at least make their face look younger again. It can be very dangerous if you expose your respiratory muscles like the diaphragm to this botulism toxin, because it can cause you to stop breathing, because you can't breathe without muscular contractions. Curare is a plant poison used by South American Indians on arrows and blowgun darts. It causes muscle paralysis, not because you can't release acetylcholine, but because it blocks the receptors for that acetylcholine, inhibiting sodium ion channels, which means same thing, the muscles can't become excited and thus can't contract. Cool thing is that we in medicine use derivatives of curare during surgery to relax skeletal muscles, which is cool. Pesticides or organopesticides, they're sometimes described, bind irreversibly to acetylcholine esterase, the enzyme that degrades acetylcholine, which is exactly what a lot of nerve agents do, which causes acetylcholine to just kind of go uncontrollably, meaning that the muscle fiber is just going to be continuously excited, and then the muscle just contracts, and so it can't relax, and eventually can be very dangerous. There's a very prominent movie scene in a movie called The Rock that came out, I think it was in the 90s, starring Sean Connery and Nicolas Cage, where they're dealing with nerve agents and they talk about what can happen in the body. It's kind of gory, but it's kind of cool. But these two things, nerve agents and organopesticides, can work the same way. And then finally, myasthenia gravis is an autoimmune disease in which a person's body produces antibodies that bind to the acetylcholine receptors in the muscle cell, preventing normal interaction between acetylcholine and the receptors, essentially doing the exact same thing as what happens when you are exposed to curare. So it can be very dangerous, and unfortunately can be deadly. This next slide here, I'm going to talk about it, but I'm not going to ask you to know it on the exam, because it's really kind of more physics related stuff and it's super confusing. But when we produce movement using our muscular contraction, our bones act as levers and the joints function as fulcrums of these levers. Lever is a rigid structure that can move around a fixed point. A fixed point is what a fulcrum is. A lever is acted on at two different points and two different forces. The effort, or E, which is what our muscular contraction is going to produce. And then the load, represented by L, or resistance, is what opposes the movement. So like when you're lifting something up like this block here, whether that's a dumbbell or just something you're picking up. That's the resistance or the load. Contracting the muscle produces the effort. And then where your joint is that's moving, that's your fulcrum. Okay. So effort is force exerted by muscular contraction. Load is typically the weight of the body part that's being moved. Sometimes that's increased when you're lifting something up. Motion occurs when effort applied to the bone at the insertion exceeds the load. We classify levers into 3 types based on the position of those fulcrum, effort, and load structure parts. The first class levers are shown here. An example being like when you tilt your head back, where the fulcrum is in between the effort and the load, the effort being the back of the neck muscles that are contracting, the fulcrum being the joints in the neck that are moving, and then the load would be the weight of your face and skull pushing down against the effort. The second class lever system is where the load is between the fulcrum and the effort, like going up into a tiptoe position. And then the third class levers, which are the most common lever systems that our body uses, is going to be where the effort is between the fulcrum and the load, like with the bicep being contracted, motion occurring at the elbow with the fulcrum and the load being out there in the hand, where you're picking something up, depending on how heavy it is, how much effort it requires. Okay, and then the very, very last thing I want to talk about is just what happens to the body when you train it with repetitive exercise. Since I have a background in exercise and kinesiology, I always kind of try to touch on that whenever possible. And this is an example of a time where I want to, and I will. So reading through, regardless of whether the training program is endurance exercise or resistance exercise. So, whether you're doing CrossFit or you're just lifting weights or you're just a runner, exercise-induced adaptations that occur in muscle fibers result from an increase in the amount of specific proteins. So training, no matter what it is, causes increases in muscle proteins, especially if you do it repetitively. A stress of exercise or the stress of exercise stimulates cell signaling pathways and contracting muscle fibers that switch on transcriptional activators, which means that they turn on specific genes to synthesize new proteins. So exercise results in transcription. Transcription, if you remember, is where we take DNA and we make RNA from it, and then we take RNA to make proteins. And these proteins that we end up making, as you can see here after weeks of training, are going to determine the muscular characteristics and its ability to perform specific types of exercise. So by producing more and more proteins, our muscles as a whole become better adapted. And a single bout of endurance exercise is not going to be sufficient enough to produce large changes in the proteins within a muscle fiber. But as I said, repetitive is most important. One exercise session does promote disturbances inside of the homeostasis that, if you repeat it over time, the result in the specific exercise-induced adaptations associated with long-term training. So that's a long-winded way of saying that exercise repeated on a daily basis has a cumulative effect, leading to a progressive increase in specific muscle proteins that overall improve muscle function. So you continuously stress the muscle, we end up producing more proteins. More proteins helps our muscles function better as we exercise. And that is just what's taking place inside the muscles. Obviously, other adaptations throughout the body take place, which we'll talk about, changes to the cardiovascular system, how our heart rate, our blood pressure, and those kinds of things change, as we'll get into those areas later on the semester. All right, that's it for muscular physiology. Thanks, guys.